this lab, I think it's a good lab because it really does make you think about what your kidneys are doing. Again, a lot of times people think of kidneys or the urinary system as just excreting waste, which it does do. It gets rid of things that your body is trying to get rid of. But I think more importantly, it's important to think about the urinary system as homeostasis, water balance, salt balance, pH balance. And so let me check. Let me go back into my module here. So except for one person, everybody has turned in their pre-lab. But most of them, if you noticed, I didn't give you the full credit, the 1 point. I gave you like a partial credit because it's partly there, but not completely. For most people got the objective, although a couple of people still, I think, didn't quite remember what objective means. It's just, it needs to be a question. It needs to be involving your explicit independent and dependent variables. What is the relationship between these independent variables and these dependent variables? What is the effect of blah on blah? Or how does independent variable affect dependent variable? So in terms of the hypotheses, some of them look great. Some of them looked overall good, but still didn't have a really explicit reason for each thing of why you expected what you predicted. And sometimes the predictions were a little vague. Like I expect the volume to be normal. There is no normal really, because what's normal depends on like what they need to do. If you just, and the amount of water people are drinking when they're in this lab, it's not at least for me, it's not a normal. It's a huge cup, way more than I'd ever drink in a single drink. Or if somebody said more or less, but didn't say more or less than what? Like the only thing you can really do is talk about the different groups in relationship to each other. Like I think the volume in this group is going to be more compared to this other group rather than increased or decreased, because that's saying that you're some starting point that we're talking about. There is no real baseline here, right? There's no baseline. It's just we have a bunch of different inputs, a bunch of different outputs for the 4 different groups, and we're going to compare what happens between the groups. So what I want to do for this first part here is just, how many people we got? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. We have 13 people. So I'm gonna put people into breakout rooms again. And what I want people to do is kind of reach a consensus, in your, like I find that the more you can explain something to somebody else, the more you really understand it yourself. I want everybody in your group to agree on what you expect and why. There's going to be kind of 12 things, right? There's going to be group 1, 2, 3, and 4. For each group, there's going to be volume, NaCl, and specific gravity. So for each of these things, you should have a prediction. It should be in relationship to the other groups. And there should be a good reason why that makes sense. And sometimes, again, it's usually just like about balance. Like you've taken in more water, so I'm going to have to release more water in order to maintain my water balance. right? But I want that to be explicit. Like when people just kind of write it and I'm never quite sure, do they know what they mean, but and they're just assuming like obvious, I don't have to say it, it's so obvious. Or sometimes people don't say it because they're actually kind of missing the point. So I want people to be kind of more explicit in this. It's just so it's really clear that everybody kind of is thinking about this in terms of, you know, water balance or electrolyte balance. And specific gravity, in general, specific gravity is going to be related to both of these, in that if you have a high concentration of salt in your urine, what is that going to mean about the specific gravity?
It'll be greater.
It's going to be high, right? Because it's a measure of how much solutes. So typically, if you have a large volume of dilute urine, it's going to be low specific gravity. If it's going to be a low volume of a lot of solutes, it's going to be really high, and then somewhere in between if you're somewhere in between. So I am going to, I'm going to put people into some breakout rooms. And what I would like people to do is adjust your hypotheses, or in some cases, for some of you, your objectives as well. And just make sure everyone's on the same page. And then we'll come back like in 15 minutes and we'll actually like look at the actual lab and the data. But before we actually do that, I really want to make sure everybody, because we're not going to actually do the lab. Normally, most of today's lab would be just sitting around for 2 hours and peeing and measuring things. And instead, it's just going to be like magic. Here's the spreadsheet of the data that comes out of it. But what's important is that you have the background to actually make sense of what it all means. So we're going to go into breakout rooms, and I will see you all in about 15 minutes. So how are people, with any questions about, after kind of discussing it amongst your groups and stuff, any questions about any of the conditions?
We were up for group 4 for the salt concentration. We were kind of on opposite sides. So we're wondering about that one.
Right. So I was talking to another group about that. Part of the problem there is you guys don't have enough information about what is the physiological effect of caffeine, unless somebody did some Googling or something, or some literature searching. There is a kind of a pop culture idea of caffeine being a diuretic, or some people will talk about, oh, you're drinking coffee, it's like you're not even hydrating because you're just going to pee more, which is actually absurd. you can hydrate just fine on coffee. But that being said, it does seem to have slight diuretic qualities, but pretty slight. I mean, it's not like coffee makes you poop. That's a pretty clear one. You can go into the literature and find real definitive things about increasing intrarectal pressure from caffeine. So that idea that you've got to poop after you drink coffee, that is supported by the scientific literature. But the thing about the diuretic quality, we'll see what happens when we look at the data in just a few moments. And the data will also kind of give us a little more idea even on the mechanism of why it might be at least a weak diuretic. It's not like alcohol where it's blocking your ADH and making your kidneys lose more water in this direct way like that. All right, so taking a look here. La, 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 la. Yeah, so if I would, yeah, so after we're done with this stuff, if you could please, like, yeah, resubmit your improved version. But what we're going to do now is go and take a look at what actually happens in this lab when you do this.
Can I ask one more question about the pre-lab or the predictions?
Yeah, for sure.
Since I submitted mine too late to get feedback,
Between our group, I did give you some feedback.
Oh, I missed it. I'm sorry. I will go check it out. Between the members of our group, we were not sure if I missed a part where you kind of wanted us to rank the groups one against the other, like which of the four will have the highest volume. Is that something that you wanted, or did you? I know you wanted to discourage us from using the word normal because that's so relative, but
right. So exactly. So yeah. So kind of relating them. So it would be, which one do you think will have the highest volume, or maybe there'll be ones with similar volumes as well. Okay. But in fact, yeah, so that should be in all of your hypotheses. It should just be relative. Like this will be the lowest or the largest, or this one will be similar to such and such. But yeah, there is no normal, so to speak.
Okay.
Okay. So I'm going to share. Okay, so this first thing is just some background information where Becky kind of lays out, because I do think before you actually see the data, it's actually useful to actually get a reality check on exactly what people were doing to actually get this data. So we'll show you, one is kind of a background of the experiment and what it looks like, what the cups look like that people are drinking out of, so you can actually get a sense of what is 800 mils really look like. And then the second little video actually shows the actual way we measured the sodium chloride concentration, the actual way we measured the specific gravity, which will actually be important when you're looking at the data, because the specific gravity requires a certain volume of urine the way we do it here. This is going to be different than we saw with the multi-sticks. So we'll just be like 8.5 minutes of videos and with some more commentary. And this also says the salt tablet can dissolve slowly. Yeah, it says, let me start this. Okay.
We're going to be covering the kidney regulation of Osmolarity lab today. And in this lab, we have students sign up for one of 4 groups. Group 1 is to consume 800 milligrams of water and nothing else. I'm sorry, milliliters, the liters of water. And this cup with this red line is 800 milliliters of water. And so we want those students in group 1 to drink that as quickly as they can, as the start of the experiment. Group 2 is going to also drink 800 milliliters of water and consume 1.5 grams of salt. And here we have salt tablets. Each tablet is 1 gram. So those students would take 1.5 tablets, and they should swallow them and not chew them. Yeah, they have.
So this is a big thing. Like when I was first like coming up with this lab like 20 years ago or so, I like didn't have official salt tablets. I'm like, I'll like gel cap some salt and experiment and see how it works. Which in retrospect was a horrible idea. What do you think is going to happen if all of a sudden you just have a whole, and I was using like, I think I had like 5 grams of salt that just instantly just unleashed in my stomach. what is going to happen if all of a sudden it's super salty in your stomach all of a sudden?
Well, you're probably not going to feel too great for starters.
You're gonna hurl, man.
Water will
The water is going to be attracted into it.
The water is going to be attracted, osmosis. So all of a sudden, water comes in, is a really high osmotic gradient. So there's a very high osmotic rate, and water rushes into your stomach, distends your stomach, stretches it out. And at that point, your stomach is stretched out, and that is a signal for hurling, for vomiting. I can tell you exactly what it felt like. It was like all of a sudden, it's like, oh my God, I felt saliva just pouring into my mouth. It's like, what the fuck's that? It turns out, I learned later that it is a feed forward mechanism to protect your teeth from the acids of vomiting. So before you vomit, your body sends in saliva into your mouth to try to protect your teeth from erosion from the acid that's about to come swirling out past them, and then I had barely enough time to run out of the classroom. Luckily, we were in the old science center where the outside was just a outer hallway, and just spewed all over the place. So these salt tablets are designed to dissolve slowly. So as they dissolve, the water can equilibrate and the salt dissolves. But it's why if you are on, in a lifeboat on the ocean, you cannot hydrate yourself by drinking seawater, right? If you try drinking seawater, as soon as you drink the seawater, you've got this really salty stuff inside you, and the water comes in and descends your stomach, and you just vomit it all out. So, you cannot hydrate by drinking ocean water, for that same reason. So, we used to use more, even with the salt tablets. What do you think it's going to feel like if you take very high levels of sodium into your body?
Well, the obvious answer is going to make you really, really, really thirsty.
Yeah, so in fact, one of the triggers for the thirst center is plasma osmolarity going up. So, over the years, we've been adjusting the levels of salt. So we see some effects, but try to make it not totally uncomfortable, where someone spends 2 hours just like wishing they were didn't agree to be in group 3 particularly. So
Plenty of water to swallow them. Group 3 is going to ingest 80 milliliters, and the red line shows 80 milliliters of water.
you can see they basically have just enough water to swallow their salt tablets, but they're not really adding in much extra water at all.
And the same amount of salt, 1.5 grams, so 1.5 tablets of swallowing the tablets, not chewing the tablets. And in group 4, they're going to consume 800 milliliters of water and 1 caffeine tablet, which is 200 milligrams of caffeine. It's like vibrant. So each group should ingest that as fast as possible, as comfortable as possible. And then that'll be times 0. And then after 30 minutes, students will empty their bladder in various containers in using the restroom. And then they are going to take.
But notice this is not going to be a clean catch. This is catching every last drop because we want to see the total volume. And it's at a half hour, whether or not they feel they need to pee or not. It's like I was mentioning, your bladder can actually expand up to a couple of 100 mils without you actually triggering that stretch reflex where you feel the need to pee. So you try to pee, usually you can. And even if you can't pee, that's data. What is the volume of that particular data point if they can't pee at all?
0.
0, right? 0 is a number. So 0 would mean they didn't pee at all.
Various analysis on their samples. They're going to measure their volume, and we have different sizes of graduated cylinders to hopefully match closely what the overall volume of urine that was collected every 30 minutes. Specific gravity, we'll show that in just a second. And also sodium chloride concentration, we'll show that next.
So there you can see the setup. It's like, and you try to put the water or salt or caffeine in your body in as much as possible a short period of time. So it's like time equals 0. Obviously, you're not going to get the entire cup of water in an instant. But you try to, like, say at time 0, that is going into the body. And then over the next 2 hours, what is going to be happening to their urine in response to that initial water, salt, and caffeine. This next video shows the actual mechanics of how we're measuring the dependent variables. So I think it's useful for seeing, so you know how we actually got the data that you're about to see in the spreadsheet. All right, so I've been in group 3, which was just drinking 80 mils of water and taking 1.5 grams of salt. And this is a half hour after, since I did my initial voiding of my bladder and taking my treatment. And here's the amount of urine that I've got. Again, I'm collecting it every last drop, because I care about the volume. So this is not a clean catch like you were doing in the urinalysis lab. You're going to collect from the beginning to the end of the flow. And first thing I want to do is figure out the volume. And I can look here, roughly, it's less than 100. So when I think about which one of the cylinders makes sense, I'm going to probably take the one that's 100. This is going to get lost if I put it in one of these bigger ones here. I'm going to pour it in. And I can go, and I can see it's about 31, 2, 3, 4, 5, 36 mils. And you don't have to keep track of my urine sample here. This is more just to show like a representative urine sample. So to measure the volume, we literally just put it in a graduated cylinder and measure the volume. And it's really this, when you have everybody in lab doing this all together, when you're actually in the room, it doesn't smell like anything, because we talked about olfaction adapts pretty quickly. But if you've been out of the room and you walk back in, it totally smells like you're in the bathroom or something. It's kind of hilarious. That is the first part. I'm also going to want to measure its specific gravity. The specific gravity, I'm going to use this urinometer. It's basically a little vial and this float. It's kind of a hydrometer, like if you were making beer. It's the same idea. And depending on the density of the urine, this little float will float further up or sink further down, and the level of the urine on this little scale here is going to tell us the specific gravity. So typically you add about maybe 2/3 of the height here of the tube. If you did not pee at all, you're not going to be able to get a value. And again, if you didn't get enough urine to actually float the float, you would just have no reading for specific gravity. It wouldn't be 0. 0 is not valid. It would just be null. You don't have a value. But I have enough urine. I should actually take a little aside here. This is kind of an important point. Like if I measure, let's say I try to pee and I don't pee at all, I can still measure the volume. The volume will be a number, 0 mils. I should say 0 mils back. But if I don't have any pee to actually float the float, I can't get a value for the specific gravity. The specific gravity is just going to be null set, nothing. I don't have a value. When we do this for real and people get no value, sometimes they say, oh, it's 0. It's not 0. 0 is a number. And in fact, specific gravity can't be 0. Specific gravity is always somewhere near 1, 1 or a little above. So make it clear that you understand the difference between not a value versus 0, which actually is a number. It means I measured it and then value was 0 versus I don't have a value for it. Those are very different things. So anyway, continuing on with my little thing here. I'm gonna now put my float in, make sure it's not gonna overflow. It looks good. I'm gonna give it a little spin. I'm gonna put it down here. Just kind of little style. You give it a little spin as you drop it so it doesn't stick to the walls. And then as it settles in, I can look here and it's about 1. So about 0.028 is my, or 1.028 is my specific gravity for this, is my. So going back to this little picture, what would this look like if my urine was less dense, it was closer to water?
The float would be further down in the tube.
Exactly. The float would be sinking deeper because if the urine is less dense, then the float won't float as high and the level of the urine would be higher up here because this whole float would be lower. What else would we probably notice if my urine was less dense?
It would probably be lighter in color.
Probably be lighter. In general, the color of the urine, not necessarily, like I said, urine can get colored by vitamins and foods, but in general, if your urine is darker color, that usually means, you have more solute per volume. Whereas if you are peeing out larger volumes of dilute urine, it tends to be more light colored, right? So, and if my urine was even more concentrated, this could float up even higher, right? So depending on how high the float goes, that is how you read specific gravity using this technique. And so this is another very common way to measure specific gravity of urine is by using this little urinometer, putting this little float. The further down it floats, the more dilute, the higher up it floats, the more concentrated it is. And then you get an actual numerical value by looking at the number on the little stalk on the float there. So about 0.028 is my, or 1.028 is my specific gravity for this. I would mark that down. Then the last part of this is I'm going to get the sodium chloride content of my urine. For that, I am going to need a little test tube and a dropper here. And so basically I'm going to need 10 drops of my urine. And it matters that the drops are actual, like well-calibrated drops. This is a quantitative test and it depends on numbers of drops that you're adding of different reagents. So it matters that I'm careful to add exactly 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 drops of my urine. Then I'm going to add exactly 1 drop of this potassium chromate solution. And now I'm going to add silver nitrate solution drop by drop by drop. And this forms a precipitate. And you'll see as a drop hits into this solution, it's going to turn brown for a moment, but then will probably become yellow again. And I'll keep adding drops of silver nitrate until this solution here stays brown. And the total number of drops that I add here are going to be the value of the total milliliters per, or milligrams per milliliter of sodium chloride that I have in there. And again, you can see it's yellow when I start. Does this help?
Yes.
Okay, I'm gonna add in a drop. You see little brown show up? And then 2, 3, 4, 5, 6. I need to kind of look at it. Still looking 7, 8. Oh, not still, it's not staying brown. 9, 10. Not quite yet. 11, 12. Ooh, it's almost there. 13. One more. 14. That's brown. So 14. It took 14 drops. So that would mean I've got about 14 grams of sodium chloride per liter, or 14 milligrams of sodium chloride per liter in my urine, which probably makes sense given that I'm probably pretty dehydrated being in group 3. Okay. So are there any questions about how we get the data? So those are the dependent variables, right? Measure the volume, measure sodium chloride in milligrams per milliliter using this little colorimetric assay. And then specific gravity using the little float, the urinometer. And so now what we're going to do is actually look at the data. The data is available for you in this same module, just as an Excel spreadsheet. So we'll look at it together right now, and then you'll do the analysis on your own in terms of comparing to your hypotheses and your final discussion and all that. But let's do that. Whoop, where to go? Where? Oh, there it is. Okay, let me. You. All right, so we have basically group 1. Again, at time 0, they had to pee, but that's kind of just emptying their bladder. So their bladder starts with nothing in it. But we don't really care what the data is in terms of the measurements here, because it's just going to depend what they've been eating and drinking before they started the experiment. So this isn't really telling us anything useful. What we care is what are their kidneys doing after they put the water and salt and stuff in their body. So we have measurements of volume, sodium chloride concentration, and specific gravity at half hour, 1 hour, hour and a half, and 2 hours after time equals 0, which is where they take in their treatment.
I don't know if I'm the only one, but I can't see the spreadsheet that you're showing. I just see the
No, I see what.
canvas.
Okay, hold on. Yeah, no, I actually, thank you for saying that. It was just a file and not the screen. Now you can see it. So here you can see group 1. Here are at time equals 30, 60, 90, 120 minutes. And here are the values. And down at the very bottom are the averages here. Group 2, we have the data. Again, let me. So notice for some of these, like this person here, there was no specific gravity. That's because there was not enough urine to actually float the float. So we did not actually, put a 0 there or anything, because that would just totally weird out the average. Group 3, this was the group that has very little water and lots of salt. We can see by the end, the average volume is really low, like 18 milliliters. Lots of people are not having enough urine left to actually get a specific gravity. Like compare that to group 1, like on the fourth P, the average volume is still around 130 mils, whereas in group 3, the average volume is about 18 mils, which is kind of what hopefully you all are expecting. If you don't drink much water, your kidneys are going to have to hold on to the water, and you're not going to lose it in your urine. Group 4, here's the data for group 4. So now what we're going to do is look at the different graphs. So what are we seeing here? Does this make sense? We can put this down. We can ask, what were, I don't know if anybody wants to volunteer. What was somebody's predictions for the relative volumes between the different groups?
We thought that the group with caffeine, group 4, would have the highest relative volume. And it's interesting, I was just looking at the curve, you can see that it's kind of late in kicking in. It stays high longer, but it doesn't peak as quickly as groups 1 and 2. And I don't see it now, but you had it on the screen for a second.
Right, and what about the other ones?
Okay, so you're asking what we predicted for volume? We said that group 4 would be the highest, group 1 would be next, group 2, and finally group 3 would obviously be the lowest.
Right, and so if we take a look, and again, we should, hopefully in addition to that, you've got the Y's in there, but
we did, sure.
So if we take a look here, we do see group 3 by far has the lowest volumes, right? Again, this makes sense. You didn't drink much water at all, and you are gonna not pee out much water, because that's what your kidneys need to do to maintain your blood osmolarity. Particularly, these guys had salt as well, so that's another reason why you're not going to be, you've taken salt in and no water. It makes sense that your body's going to hold on to the water to keep your blood osmolarity in a good zone. Whereas everybody else who drank in lots of water, just as a group, they all peed out a lot more water, which makes sense. If you take in a lot more water, you're going to have to pee out more water to maintain balance. Relatively, though, the group that took in the salt with the water, doesn't have quite as much, which actually also makes sense, right? If you have extra salt, you're going to have to be more careful about just letting go of water, because your osmolarity has to deal with the salt as well. So you're going to have to probably be getting rid of water, but also salt, so the water's not going to come out as fast. Like the salt actually helps keep the water in the body. I would say groups 3 and 4, they look pretty similar. And again, people talk about caffeine being a diuretic, and even if it is a diuretic, it's a pretty mild diuretic. It doesn't radically change the amount of pee you have compared to not having caffeine.
I have a question about that too. So I definitely looked on the internet and saw, oh, it's a diuretic, and here's the mechanism, and it's a good reminder to look side-eyed at the internet, but also it would increase your blood pressure a little bit, right? Caffeine.
Well, so that's interesting. I mean, if you're thinking about a diuretic, that would be the opposite. That would take it down by reducing your blood volume.
Not volume, but pressure. Oh, I see what you mean. Yeah.
But then it could also, it's a stimulant, which could increase your heart rate and things like that. So that could be increasing your blood pressure. So it could be doing either, depending on how what point of view you're looking at. Yeah, I'm not actually sure if what its actual effects are on blood pressure, if it has much effects on blood pressure in the big picture.
So it's okay if we talk about it being like a diuretic in our like pre-lab and our predictions, but in the discussion, should we tie it back to like how it's only a mild
Yeah, I mean,
diuretic boost?
I'm looking at here, the reality is it's not radically changing the urine output compared to just drinking pure water. So even if sure is a little more pee here than there was in this case, but it's not that impressive. If we look at NaCl concentration, we're gonna, let me get this a little smaller. Here we see concentration for group 3 is very high compared to everybody else. And all the other groups are pretty low. Although you do see group 2 is a little higher than the other groups, which makes sense because in addition to getting rid of water, they're also getting rid of salt. And the other thing, it's interesting looking at this group 4, the caffeine group actually seems to have a bit higher salt coming out, which could make sense. If your body is getting rid of more salt, the water is going to follow the salt, which is going to cause, more water loss. The other thing that I did for your graphs was this is salt concentration, which is, salt per volume. But it's also interesting to look at just what is the actual amount of salt. If you just dried out the pee, how much salt would be there in the beaker?
Mhm.
So to do that, you just have to multiply. It's like milligrams per mil times how many mils of urine is there, right? So this is going to be NaCl amount. So this is actually actual amount of salt that's leaving the body. And this is actually kind of an interesting thing to pay attention to. One is group 3, even though it had the highest concentration, notice it actually has the least actual amount of salt that's leaving. Why do you think that is?
Because your body's trying to retain water, so it's keeping the salt inside.
So there's a mix. One is that if you're trying to conserve water, you often do pull salt back into your body in order to keep pulling the water back into your body. And the other thing is you have to have a minimal amount of volume to actually carry the solutes out of your body. Like groups 1, 2, and 3, 1, 2, and 4 have lots of water to dissolve whatever you're trying to get rid of. Groups 3 have very little volume coming out in order to dissolve whatever it's trying to get rid of. So even if you're trying to get rid of it, you're kind of maxed out by how much water you actually have to dissolve it in. So even though the concentration is actually quite high, the volume that's actually leaving is pretty low. So you're not carrying as much out. That's when I was talking about water balance, that you have to have some minimal amount of urine production, even if you are trying to conserve water, because you need some volume to actually dissolve stuff in so it can actually leave your body, right? It can't just like disappear. Okay. But I do think if you look at group 4, the caffeine group, so when I actually looked at some of the stuff on caffeine, I think caffeine does seem to cause a little more salt excretion, which would then follow that it would cause more water excretion because water is going to follow the salt. So caffeine seems to have some effect by increasing salt excretion and therefore some water excretion. But again, it's not dramatic. It's not like, It's not like, and then you can see group 2, like the difference between group 1 and group 2 was only salt, right? They both had 800 mils of water. And we can see group 2 that had more salt, definitely had more salt excretion compared to group 1. So that makes sense. They had, you put more salt in, more salt came out to maintain balance. So that's pretty intuitive there. In terms of specific gravity, if you think about salt concentration, specific gravity is basically going to follow that pretty much. Group 3 that has a lot of salt is going to have the highest specific gravities. Group 2 that ate the salt and we're having to get rid of salt had a kind of this low, but a little higher than the rest. Group 3, which was the pure, group 1, which was the pure water, had the lowest specific gravity because it's just getting rid of water and no solute. And group 4, which is getting rid of, it seems like excess, a little extra salt compared to normal, is going to have a little higher. So this should also be kind of intuitive. Does that make sense? So it's kind of nice. It's nice to see that. And it also kind of hopefully gets you out of this thought of normal for urine. It's like, there's not normal. It's just like, what do your kidneys need to do to maintain water balance and salt balance? That is what the composition of your urine is. The pH of your urine is going to be, what does it need to do to maintain pH balance? Do I need to pee out acids or hold on to them? So that's kind of that. Are there any questions about this data? No? Okay.
Oh, you want all of that data in our lab book, right? Every chart.
Yeah, no, for sure.
Okay.
For sure. All right.
Sorry, one more question. The graphs, sure. All the specific numbers too, or just the general graphs?
The numbers are kind of reflected in the graphs. So, like if there were kind of outliers and there were things where you were specifically addressing things in the raw data that was coming into your discussion, then you would have those. But for your discussion, you just need to have the averages and the trends. So then that's just reflected in the graphs. You don't have to have all the raw data in there. What's become more of a thing in scientific publication is having online supplementary materials. So now a lot of times when people publish papers, they'll have kind of the results that are most important for their discussion. And then they'll have a website where you can go and get lots of more nitty-gritty details if you're interested in looking at the kind of finer details and kind of looking at the uglier stuff under the hood, so to speak, of a data analysis. So having supplementary materials now is very common in scientific publishing. And again, you can do that now easily because data storage is so cheap. So these journals can just store buttloads of data and not worry about like running out of storage space or paying for cloud storage or whatever. It just stays in the cloud forever now, or until the fall of civilization. So that's that.